Hi, my name is Carmen and I will be your guest lecturer for Biochem 108, where biochemistry is for everybody. Today we'll be discussing a fascinating mechanosensitive ion channel located in the plasma membrane of cells and functions as a receptor that senses mechanical forces like pressure and stretch. Piezo 1 can be expressed in various cell types, including endothelial cells, red blood cells, epithelial cells, and contributes to functions like vascular development, blood flow regulation, and touch sensation. When activated, it allows cations such as calcium, sodium, and potassium to flow into the cell. This protein is crucial for processes like cell adhesion, blood vessel formation, muscle development, and fluid pressure detection. Piezo 1 has a unique 3D structure. It is a propeller-shaped homotrimer with 3 blade-like subunits. Each subunit contains a peripheral blade with 38 transmembrane helices, a C-terminal domain, an anchor, and a 90 Armstrong long beam on the intracellular side. The central pore is found in the inner helix and cap domain near the pore and controls ion flow and channel inactivation. The 90 Armstrong long beam is a long transmembrane alpha helix that connects the blade to the pore, potentially acting as an optimal lever for controlling Piezo 1 activation and transmitting mechanical forces. The peripheral blade is curved, flexible outer structure of Piezo 1 and is in charge of detecting membrane tension and triggering channel opening. The normal protein exists in a curved resting state with blade angles around 130 to 150 degrees. When the mechanical force is applied, the blades flatten to around 180 degrees, opening the ion channel. This is a pipe cleaner model I made of the Piezo 1 protein, complete with a central pore region, peripheral blade, and a 90 Armstrong long beam, to help you all visualize its structure. The pipe cleaners demonstrate the primary structure as the amino acid sequence, while the twists and folds represent the secondary structures like alpha helices. The 3D shape of each subunit forms the tertiary structure, and the full 3 subunit ion channel represents the quaternary structure. Now let's take a look at how the gain-of-function mutation alters Piezo 1. In this model, I've used beads to represent amino acid changes that disrupt its normal structure and function. Mutations in the central pore prevent proper inactivation, keeping the channel open longer and causing red blood cell dehydration, constant ion leakage, and increased fragility. Bead mutations disrupt the structural support for gating, further contributing to uncontrolled ion flow and dehydration in red blood cells. These amino acid mutations reduce the resting curvature of the protein, shifting the blades to a more open state at baseline. Instead of maintaining its usual 130 to 150 degree at resting angle, the blades are now moved closer to a 160-180 degree, making the channel more likely to stay open. Upon exposure to mechanical stimuli, non-mutated Piezo 1 channels undergo activation, followed by rapid inactivation resulting in a prompt closure. This mutated structural change caused the protein to close improperly and result in leaky ion channels, even when no mechanical force is applied. So how does this structural change affect function? Even a small reduction in a blade curvature degree can significantly impact Piezo 1's response mechanisms and increase its open probability, leading to excess ion flow and disrupting cell homeostasis. The clinical impact of this persistent activation leads to increased ion permeability and excessive calcium influx, and in red blood cells, this triggers potassium efflux and water loss, causing cell dehydration and hemolysis. This mechanotransduction defect underlies dehydrated hereditary somatocytosis and other Piezo 1 related disorders, showing how structural mechanics directly influence disease pathology. In DHS patients with Piezo 1 gain-of-function mutation, red blood cells are dehydrated due to chronic activation of KCNN4 and typically show increased deformability at lower osmolality values, confirming changes in the water content equilibrium. This is the basis of DHS, where fragile red blood cells break down more easily, leading to anemia and circulation issues. At the organism level, this can cause fatigue, jaundice, and complications related to blood flow regulation. While my pipe cleaner models may help visualize Piezo's unique function and shape, they do have their limitations. First, it does not accurately depict the protein's atomic level structure, as real proteins have complex folding patterns and intricate bonding. As you can see, there is only so much you can do with pipe cleaners. Second, the model lacks the fluidity of an actual membrane-bound protein. In reality, Piezo 1 is embedded in a lipid bilayer and interacts with surrounding molecules. Finally, as I hope it is evident, these piezo molecules are not to scale. While this model is much larger and simplified for visualization purposes, Piezo 1 and other ion channels alike are actually only nanometers in size. Despite these limitations, I believe the model effectively highlights key structural changes caused by gain-of-function mutations. In summary, Piezo 1 is a crucial mechanosensitive ion channel, but gain-of-function phenotypes can cause a series of reactions, including excessive ion flow disrupting red blood cell homeostasis and causing disorders like DHS. Ultimately, understanding these structural and functional changes are important in helping researchers explore potential treatments for mechanosensitive disorders. Thanks for watching, and remember, quizzes are due Tuesday. See you next week!